welcome to today's webinar. This webinar is on the truth about bacterial and enzyme products. And truth is kind of a big claim here, but in general, there's been a lot of misinformation around about bacterial and enzyme products and how they can apply to wastewater systems. So the point of this is to dispel some of those myths and give people some sense for when it is appropriate to add a bacterial product and how to sort through marketing stuff and figure out if a product is actually likely to work in your system. I'm a chemist at Aquafix. My name's Dan, and I've been here for a while. Aquafix Laboratories, we do a lot of work with customer consultation, microscopic observation, and treatability studies for clients. If you have any questions about our lab services and how they may apply to you, please get a hold of us and let us know what sort of services you'd be interested in there. We also do most of our product development work in this lab, as well as producing product literature so that we have some data on your product. You'll see throughout the webinar, data is one of the keys for determining if you have a product that has any hope of working or not. We really like to see products, particularly for bacteria and enzyme products, to have data. First, we're going to sort through bacteria and enzymes and go through some myths and misconceptions. We're also going to be covering how bacterial and enzyme products work, and we'll get some focus points to make sure you know how to apply them appropriately to get best results. We're also going to go through the data we have on several of our products. Now, I won't be covering every product that is bacterial-based that we offer. There's quite a few of them during the webinar. But if you're curious about a product that hasn't been heavily mentioned, please ask us, and we will have some sort of literature to give to you on it. Just a quick overview. We have our bacteria and we have our enzymes. They are two separate categories, but there's a couple keys which we'll come back to throughout the presentation. The main one being bacteria tend to work more slowly, but they can reproduce, and enzymes tend to work more quickly, but they cannot reproduce. Enzymes in particular require a high concentration of target substrate to be very effective, whereas bacteria can be more effective in lower substrates with supplementing diversity to take care of specific roles in the system because of their ability to move and reproduce. So this is just a fun little size comparison here. You can see we have our E. coli bacteria, which is a pretty good size bacteria, and we have it next to an enzyme. And you cannot actually really see the enzyme on your screen at all, probably, unless you have a very large display. It just looks like a little speck of dirt. And that's a pretty representative size comparison of a bacteria versus an enzyme. We occasionally get questions about whether we saw certain molecules or enzymes under the microscope when we're doing microscopic analysis. Just to be clear, that is not generally possible. They are thousands of times too small to show up in a standard light microscope. You need to have much more powerful equipment to be able to see an enzyme at all. All right, so bacteria. Interesting note about bacteria. Particularly in municipal systems, you have a lot of bacteria entering in the system from things like human digestive tracts. So fecal matter going into wastewater plants contains very high levels of bacteria, but these bacteria don't maintain any presence at all in like an activated sludge system. They basically all go away. They fail to thrive in the system. And this is something you have to think about with any bacteria, but particularly bacterial products as you're applying them. Bacteria need the right conditions in order to be able to thrive in a system. Otherwise, they need to be added repeatedly, and you kind of have to hope for the best. Many bacteria can swim to their food. We'll go through that a little bit more in detail. This helps them be more efficient at targeting substrates in some cases. But basically, remember throughout the presentation, your system has a lot of bacteria. If you're adding a bacteria, you want to have a purpose to add the bacteria. You don't necessarily want to be just adding some bugs to the system. So I wanted to briefly go through symbiosis as a concept. Generally, we're talking about interactions between organisms within a system. Bacteria have a broad range of interactions, and this is one of the reasons it's very difficult to decide whether a bacteria will thrive or fail to thrive in a system. There's a lot of different species which will be competitive but not interacting directly with the bacterial species in the system. But bacteria can also be mutually beneficial and work together, and in some cases you won't have any of one bacteria if you're missing another bacteria. A good example of this is ammonia oxidizing bacteria and nitrite oxidizing bacteria. Ammonia oxidizing bacteria produce energy by oxidizing ammonia. that gets converted to nitrite when they perform the oxidation, and then you have a substrate which the nitrite oxidizing bacteria can then oxidize again to produce more energy. If you didn't have ammonia oxidizers, you wouldn't have enough nitrite to sustain a population of nitrite oxidizing. So this would be sort of a mutualism. Both parties benefit somewhat from the relation. In regard to the ammonia oxidizers, if you have too high levels of nitrite, the ammonia oxidizers are not able to function in a system, so you need to keep those levels down. However, you can also have our basic competitive relationships, but you can also have a bacterial party that could actively harm another party present through predation or parasitic means. So keep that in mind. There's a lot of different interactions going on between bacteria in your system. So if you're just adding some unknown bacteria without any data, there's a good chance that it won't do anything, would be the most likely outcome, or it could actually do something harmful in your system. And that's going away from the possibilities of if you have an unknown bacterial bag, you could even potentially have something like a pathogen in it. You want to have a little bit of data and some assurance that pathogen levels are relatively low in a bacterial product before you apply it. So what are bacterial products? I have a couple bacterial products pictured here. The one on the right is a bug on a rope, which is one of our bacterial products we use in lift stations and collection systems. And the one on the left is our sludge RX product, which is designed for reduction of sediments. Now, these are both kind of interesting. I'll talk a little bit about both of them and some data in relation to them throughout the presentation and kind of what their goals are. But generally speaking, a bacterial product is added to supplement a system to fulfill a certain role more efficiently. The cleanest example would be adding nitrifiers. If you add nitrifiers to a system, you probably had just had an upset of some kind which messed up nitrification in your system. Nitrifiers are relatively slow growers, so it'll take them a little while to come back. Eventually, they would repopulate if you did nothing, but you can dramatically increase the repopulation rate by simply adding a nitrifying bacteria. Generally speaking, nitrifiers that are effective in wastewater are naturally occurring organisms in wastewater, and therefore you can be reasonably certain you'll have good success, assuming you're using a nitrifying product that seems to have a good history of working in numerous systems. These products can work in multiple ways. The ideal way a bacterial product would work would be if you fed bacteria into a system, they were able to thrive and persist in that system and integrate themselves in the system bacterial population, so you wouldn't need to reseed them and they'd be fulfilling some beneficial role like our nitrifiers we just mentioned. However, you could have another means which would be somewhat effective in a process, but less than ideal circumstances. And a lot of the time, this will be more common because it's hard to get a non-native bacteria to permanently take off in a system, because it wouldn't be, if it's not in the system to begin with, there's likely some reason that bacteria isn't getting a competitive edge. So bacteria can be effective in a system without fully populating. They can perform a role after they have been added into the system, and then they can stay alive in the system and continue to perform that role for a period of time. They'll likely reproduce some. Eventually, they'll get washed out of the system, so you'll need to add more, but they still have had time to perform a meaningful role. The third possibility is if you were to add a nitrifying product and have a bacterial product and have the bacteria die immediately when you add them into the system, but they release some useful enzyme or nutrient which can help to boost the system in other ways, you could say. Now, generally, the longer the lifespan of a bacteria in a system, the less frequently you'll need to add it, and the less timing you'll need to make sure you're getting the full effects of a bacterial product. So for example, if you throw in a packet of a bag of some sort of bacteria in the morning, but you have a lot of the substrate you're concerned about coming in the afternoon, if those bacteria aren't able to persist until the afternoon, you're not going to get much benefits from them. Another note is very frequently bacterial products are added with some sort of nutrient blend in the system. I have some data in a couple slides which will show you exactly how this sort of thing would work, but generally speaking, since bacteria have to compete when you add them with a whole lot of different organisms, it helps to provide some nutrients for them to get them to activate and begin to integrate themselves more rapidly in a system. Otherwise, it can be very difficult to get them to thrive. And a lot of the time, if you don't have any nutrients present in a bacterial product that are specific to the bacterial culture present, you're going to have difficulty getting the product to work for periods of time. So, what do you need to make an effective bacterial product? I think a fun way to look at this is, what do you need to do to make a bacterial product with okay literature that is also super cheap and is easy to store? So, you're looking for a bacteria in these cases that will grow to a high population and can sit on a shelf. There's a lot of products like this on the field. They're very easy and cheap to grow, but a lot of the time, the reason they are easy to grow to high concentrations is because they're not a native wastewater bacteria, which means you can't count on them working at all in a wastewater system. You can throw in as many of a bacteria as you want, like our discussion earlier about the human intestinal bacteria, and if they don't have the right conditions and they don't have a little help, they're going to fail to thrive. So you can easily make a bacterial product that is cheap and has a good shelf life with bacteria in it, but there's a good chance it isn't going to work. A lot of the time, to make an effective bacterial product, you have to have something that is somewhat native or to a system similar, or specifically a wastewater treatment system or a sewer line. Wherever you want it to apply, it is better to have a native strain of bacteria with known beneficial impacts. Some of the time, these bacteria won't have as long a shelf life or as high a concentration in the product, but overall, they will be a much better product. So I wanted to talk a bit about flagella and taxus. We've had a number of comments over the year about people thinking bacteria are unable to move and swim around in a system. And there are some cases of bacteria that can't swim, but very frequently swimming is a competitive advantage in aquatic environments. So a lot of the bacteria in wastewater are capable of swimming at least some time. So it's kind of interesting. I have a little diagram here. When a bacteria is swimming in a wastewater system, it is gradually trying to move itself towards some kind of attractant, usually a food substrate, which it is looking for. This is more of an active approach than you'll see with a lot of enzyme-based products. So your bacteria will have all of its flagella rotating in a counterclockwise motion, and then it will do a run. That's when it's swimming, all the flagella will be organized and pushing it forward. Eventually, its flagella will start rotating clockwise, and it will start to sort of tumble around in its tumble phase. Then it will proceed to do another run and keep running kind of randomly until it gets close enough to an attractant that it realizes there is something present there for it, and then it can direct itself to the attractant and make contact on the surface. So it's a very active approach, and this is why if you add a packet of bacterial product to one point in your system, it may actually have a benefit in a different point of your system. If bacteria never swam, then it would be difficult for them to migrate in this way. So another goal you might have with adding a bacterial product would be to produce better flocculation in a wastewater system. Now, slime can be kind of scary to talk about in some cases, but generally speaking, we're talking about extracellular polymeric substances, which could be slime in a wastewater system. Extracellular polymeric substances are very significant to bacterial operation. In sewer lines and collection systems, you're very likely to have a biofilm. That's an EPS layer growing on the sides of the sewer line. That's more typical of high flow environments. You might see fixed film systems. They use EPS in order to stick together. But you can add a bacterial product that contains a bacteria that are good at producing EPS. And if you have the correct conditions present, they can encourage flocculation in a system where flocculation was not taking place otherwise. So here's a little quick conceptual diagram. On the left is representing a sample we had, which was a mixed suspended bacterial algae culture we were working with in the lab. This sample was not settling whatsoever, pretty much no matter how long we set it out. So we attempted to apply a bacterial product in order to see if that would assist in flocculation, and we're comparing it to coagulants in this study. So it'll depend a little bit on what sort of conditions are present in your system and what sort of bacteria are being added, but if the bacteria are working, they produce a little slime, they can get the other non-flocculating bacteria to stick together, and then they can settle out and form floc. And this is a good application for this is nitrifying bacteria tend to be poor floc formers. they tend to produce very little EPS compared to many other organisms. So in a lot of cases, it's likely that the nitrifiers are being held in the system by other flock-forming bacteria present. But anyway, in this specific test, we added a little bit of a bacterial culture to our suspended bacteria, and after 1 week, we were adding 10 parts per million, we saw about a 20% reduction in suspended bacteria measured by absorbance in this case. And that is an okay result. We were getting some flocculation with the addition of bacteria. In this case, we did see some better results with the coagulant in this case, which is what we ended up recommending to the customer. But ultimately, this will be a case-by-case basis whether you want to use a coagulant like aluminum chlorohydrate or if you use a bacterial product to accomplish a similar goal. So here's a couple keys, but the main ones we've already discussed a little bit. Bacteria need some nutrients in order to be able to thrive for a period of time in a system. They also need proper environmental conditions. Generally speaking, if you add bacteria to a system, you don't know what effect they'll have unless you have some data showing that it's been used in other sites with success in some way. It doesn't necessarily need to be perfect data. In a lot of cases, it can be difficult to produce really good data for bacterial products, but if there is nothing present, you can be very sure that there's not a good chance of that bacterial product making a difference in your system. Also, be suspicious of very inexpensive products. Like I mentioned earlier, if they actually have bacteria in them, and in some cases it's likely they do not, most of them will give them the benefit of the doubt, assume that there is bacteria in these cheap bacterial products. It is likely something that was selected because it's easy to culture and store, and not necessarily because it's good in a wastewater system. All right, so we're going to lead off with collection systems. So collection systems, in a number of ways, are good sites to apply a bacterial product. There's very high concentration of substrates present, so you have some potential for rapid bacterial growth, but they have some significant limitations. One, there tends to be a lot of variability in collection systems and pretreatment systems with what the loading rates are. And if there's variability in the system, you need a bacteria to persist for a larger period of time in order to be able to cover the variable loading. A lot of the time, since the flow through is so high in collection systems and in many cases pretreatment systems, you won't be able to keep the bacteria in the system long enough to have it reliably interact with the substrate without very precise timing. And particularly in collection systems, you have so many different sources of waste that it can be very difficult to predict when you're going to have a period of high loading versus low loading. So in these cases, you have to be aware of how is the bacteria going to stay in a system long enough to have an impact. So I mentioned earlier the product Bug on a Rope, and this product was designed really precisely for lift stations. It has been used in some pretreatment systems and that sort of thing. So Bug on a Rope is a product designed for lift stations, and it is a bacterial block which is designed to be slow release and provide media for bacteria in a lift station or sewer line. As long as there's a lot of grease and a decent amount of flow through, the bacteria in the block will slowly begin releasing throughout the course of a month and perform some work while they're on the block as well. And they can participate in accelerating degradation of grease in a lift station so you have less grease buildup. So in that product's case, there's a specific mechanism where it is actually a solid block, so it is designed to stay in a system. Something like a suspended media or something along those lines can be helpful in this case. Obviously, you want something that's contained that isn't going to mess up pumps or something. But if you had a media block or something, it would be one potential way you could get some sort of bacterial biofilm to begin doing work for longer periods of time in a sewer line. So you need to be less precise about application times. Another good application of bacterial products would be a Greasezilla product, which it's a biocatalyst product. It has a number of elements making it work. It's kind of our hybrid between enzyme and bacterial products. But its bacteria have time to work for a simple reason, and that's because we are spraying it directly on top of grease in a lift station in ideal circumstances. If it sticks to the grease, it has enough time for the bacteria to make some impact and begin to degrade the grease present. So that would be another option of you have somewhere for the bacteria to stick so they're not getting washed away in the system. And in the case of Greasezilla, they're going to stay stuck to the grease until the grease physically breaks apart and is washed down the line and begins to be degraded. So that's the kind of thing present. All right. So Next thing I wanted to talk about is inside a wastewater treatment plant. In some ways, wastewater plants are ideal places to add bacteria. I mean, they have a lot of bacteria present, they have bacteria that are staying in the system. It must be a good site, right? Well, in a lot of cases, they are a good site, but again, as mentioned before, you have to be aware of the competitive environment in the wastewater plant. There are going to be a lot of bacterial products that may work in many different sites, but they might not work in your system, and that might not even necessarily mean it's a bad product. It may just be a product that hasn't been tested for your specific environment. So, it's impossible to be entirely certain if the product is going to work or not prior to a on-site test or at least a small scale study, but you can generally see that if you have similar plants using the product, you can make a much more informed decision. All right, so continuing on, I want to go through some data on our bacterial products. And we're going to be talking about Vitastim nitrifiers, Sludge RX, and Dynamic Duo. We also have a case study on Bug on a Rope and Greasezilla coming up in a couple of slides as well. So SummerSlam, this is a really nice illustration for how a bacterial product interacts with nutrients that could be added within a nutrient product. When we were developing SummerSlam, we were curious about if our nutrient blend was helping to improve the performance of bacteria. In this case, we were looking at pond water specifically. Pond water would be similar to a polishing lagoon. Nutrients are relatively low, but there's still a decent amount of nutrients present to supplement the system. So you can see we got very distinct results. On the base, our pond water didn't have much change in absorbance levels throughout the duration of the test, indicating there was very limited bacterial growth occurring in the system. Whereas if we added nutrients, we could see some growth taking place. You can see the red line near the bottom is sloped up slightly, and it comes close to the level of bacteria in the end of this test duration. And if you kept the test running, you could assume that the nutrients would eventually pass up the straight bacterial product. However, when we combined, so you can see the bacteria, the levels increased quickly and it stayed at a steady 0.02 for the absorbance, indicating it was basically there was not a lot of growth happening after the first day of the test or so. We basically flatlined and the bacteria were kind of just persisting in the system, not doing a whole lot. But when we add both the nutrients and bacteria, which is the top graph, the green line, we noticed persistent growth throughout the entire duration of the study, and we also had a faster increase at the beginning thanks to the addition of bacteria. So that is how a bacteria and nutrient blend will kind of work together to establish bacteria more efficiently in a system. So here's another quick study. We were doing this with quat toxicity and Vitastim Dynamic Duo. So we've done a lot of internal studies on Dynamic Duo. This one was interesting, and it was a bit of a troubleshooting experience because one thing you'll need to watch out for in your wastewater plants, if you have an issue with toxicity occurring, such as quat being fed into your system or something like that, you need it to leave the system before you can feed a bacterial product, and it can be hard to determine if it has completely left the system or has become inactive. So generally, it's best case to wait a couple days. In our study, we did this several times, and we noted it was nice to wait until 2 days after we added our quat in my lab reactor in order to have the nitrifiers be able to grow. So after doing that, you can see we added quat. This wasn't an exceptionally high ammonia feed, but we can see ammonia went up to about 6 part per million a day after adding quat. We added the Dynamic Duo on day 5, and you'll notice the control basically flatlined for a couple more days and eventually dropped off, and it started to drop between days 8 and 9, and then it dropped more quickly from days 10 to 11. But after adding Dynamic Duo, since we were populating the bacteria a lot more quickly after the toxic event, we saw near perfectly normal ammonia reduction on day 7 rather than day 11 of the study. Final thing I wanted to talk about was some of our Sludge RX testing. Now, this is something that our lake management has worked a lot with, but this is a test that's basically representative of a wastewater polishing lagoon with accumulated sludge. So you can see TR13 represents Sludge RX, and our control is with no additive present. And this product is designed to reduce sludge. Conceptually, if you're converting organics in the sediments to a more inorganic form, there will be a lot less water contained, which makes the sludge take up a lot less space. While you're degrading the sediments, you're also converting nutrients from more bioavailable forms to less bioavailable forms. So those are kind of the benefits of reducing sludge. In addition, sludge accumulation can be a problem for mechanical reasons. But you can see here with the addition of sludge Rx, we were comparing the slope of the sludge reduction throughout the duration of the study. You can see our control line is fairly flat here. We got a slope of 0.0042 as a magnitude for the slope, which is a fairly flat slope, whereas with the addition of sludge Rx, we ended up with a slope of 0.0225, and we're seeing, about 5 times more rapid reduction in sludge with the addition of this product. Now, this product is designed to be used for a long period of time. You can see here we applied it for 13 weeks, but generally speaking, this supports the idea that sludge Rx is a bacteria that can persist long enough in the system to make a difference in the organics of the base of the pond or wastewater lagoon. Just another note to mention it, you can add sludge Rx to things like if you have settled sludge in an aeration basin, it will be effective in those applications again. They just tend to not be as good an area to measure for a study because the sludge is not usually evenly distributed. So the next thing I wanted to discuss about, and is just a general note I wanted to mention, is that greasezilla and bug on a rope were tested by a company we've worked with, BAI Engineers. So we really like working with customers for field case studies. For customers who provide us data, we tend to work with them a lot because field data is always beneficial as a complement to lab data. You need to know that a product will both work in the lab predictably and in the field predictably to be really certain it's going to work. So this company took some measurements for us and provided us the data on their bug on a rope and greasezilla treatments. And anyway, you can see what they were measuring was levels of BOD and TSS exiting several lift stations in their system. You can access this full study through the attachments to get their own conclusions. But we were seeing fairly steady BOD levels in the first 4 measurements they have listed here. TSS tends to be a little bit more spiky. We saw a really high odd TSS level on the May 21st date. But generally speaking, BOD was steady. They were interested in lowering BOD and TSS because it was less expensive for them as far as discharge permits go. So they began adding bug on a rope and greasezilla in July. And you can see after that addition, we noticed lower levels of BOD and TSS in August. So they had about a month of work with those products. They were seeing decreased levels. So this is, as a case study, we tend to have a few less data points than I would generally like here, but it was really nice of them to collect the data, and it's very helpful to see that sort of thing. In this case, they decided that it was a good application for the products, and they continued to add them. Obviously, for case studies, it can be difficult with everything else going on in wastewater to take tons of measurements and data, but it is really nice when people do take the time to collect data, as in this case. So thank you to BAI Engineers. So now I want to get over to enzymes a little bit more. So enzymes contrast bacteria in a number of ways, but the key here, if we're looking at it, is enzymes are faster, like we discussed, and bacteria are slower. But bacteria are a little bit smarter. So here we have a quick video I want to show just to kind of illustrate the nature of enzymes moving around in a system. You see these large blobs are enzymes, and they're breaking down substrates. So you can see the substrates bump into the enzymes essentially in a random method, and when they do interact with the enzyme, they stick in the active site, and the substrate is broken down to a point where it's a little easier to access by bacteria. So that's kind of just an illustration. It's important to keep in mind this because enzyme products tend to work really well if there is a high concentration of substrate, and they tend to function fairly poorly if there is a low concentration of substrate because you need the substrate to bump into the enzymes. And in some cases, you actually need more enzyme to deal with a lower concentration of substrate than you would to deal with the higher concentration of substrate, which is a little unintuitive, but the idea is if you want the enzyme to contact a substrate, you need to either have high levels of substrate, high levels of enzyme, or both for it to work. So enzymes are not bacteria. It's best to not get them mixed up because one of the keys is that enzymes can be produced by bacteria, but they cannot produce themselves. When you add an enzyme in a system, it will always exit the system in the hydraulic retention time present. So if you have an enzyme, the other thing that will be the case for an enzyme is the more reactions that are catalyzed by an enzyme in a system, the enzyme will eventually sustain damage and become no longer active. Generally speaking, enzymes tend to survive a little bit longer in cooler temperatures and a little bit shorter periods of time in warmer temperatures. This doesn't necessarily mean warm temperatures are a bad application for enzymes. In a lot of cases, they're actually a great application for enzymes because enzymes work faster in warmer temperatures. But it gives you kind of a trade off when you're considering, will the enzyme be in my system long enough to be able to make a difference? Now, enzymes are naturally produced in wastewater systems all the time. Generally speaking, enzyme supplements will be exoenzymes, which are excreted by bacteria in order to break down complex substrates into a form which can be uptaken inside the bacteria and they're then degraded and used for a food substrate. They will not survive indefinitely in the system, but if you do have a decent amount of holding time and conditions are favorable, they will persist for a decent period of time. It's usually very easy to add an enzyme product at the beginning of the day in a system with decent holding time and have decent results throughout the day with that product. So, keys to these products. Temperature has a large influence on the rate of enzyme functions. Generally, enzymes still can work at cooler temperatures, but require more time to work. And this can become pretty dramatic. Generally speaking, our enzyme products we don't recommend using below 10 Celsius because their rate of operation becomes very slow. When you're looking at an enzyme product and you're thinking about purchasing it, make sure they have some idea of temperature and pH values where the enzyme will work. They must have some literature on it, otherwise you have no idea if they're going to work on your system. You need to make sure you're adding them to a system that is appropriate with that enzyme, otherwise the enzyme can totally fail to work or fail to persist for long enough to do anything in a system. All right, so couple other notes on enzyme products. Unfortunately, the word enzyme gets thrown around an awful lot as just a marketing buzz term. A lot of enzyme products may have an enzyme label, but actually be bacteria or a solvent and contain very little enzymes present. So this is something to kind of watch out for. An enzyme will actually begin breaking down a substrate, whereas something like a solvent is just going to make it into a more soluble form, especially in the cases of fats, oils, and grease, that kind of thing. And bacteria can be used in place of enzyme, but generally speaking, you won't get great results if you're trying to deal with a high concentration of substrate and you throw in some of a bacterial product for the goal of supplementing enzymes in the short term. Bacteria need a lot more time to work on that sort of thing so they can establish a population in a system. The nice thing about enzymes is they are fairly predictable relative to bacterial products. You're not relying on biology, it's just facilitating a chemical reaction. So you can usually test the effectiveness of enzymes a little better. So we do that here in a couple of different ways, and a lot of these methods could be used on site for wastewater plants. Oxygen uptake testing is a very nice, relatively simple to run method for analyzing the performance of an enzyme product. Basically, if you have an oxygen uptake test, it will increase the rate. If the enzyme is effective on the substrate, you'll get more bacterial activity because substrates are easier to break down, and your oxygen uptake rate will increase, which directly corresponds to faster bacterial performance. So you can run this type of test. We use the YSI multi-lab setup. It's not necessarily the best or worst setup. It just happens to be what we use on our site. It's nice because you can run 3 bottles at the same time. But generally, you'll use a feed substrate and some bacteria of interest to degrade your substrate present. So there's a couple keys to using this testing. It can be a little difficult to determine exactly what time frame you want the study to be. If you have too much bacteria, then the test may run too fast and you may not get a good view of the substrate you're trying to look at degrading. And there's a little bit of guesswork in that as well. So it's easier if you have a lot of experience interpreting and working with the results to determine if a test is effective, and you may need to make some changes for your specific test in order to get it to work optimally. In systems with variable loading, you need to be very careful with these tests. We've tried to collect, we've done a lot of research studies with oxygen uptake, and you have to be real careful about collecting samples because you want to have a high concentration of substrate. For example, protein. There have been a number of times where we've had samples collected and they've basically looked like tap water and they had like 20 parts per million COD in it. And it's like, this seems like this might just be some sort of pure water rinse or wash water or something like that. This is obviously not their actually substrate because you almost need to do no work in order to have good effluent quality in these cases. Make sure when you're collecting a sample for this type of test, you have a high concentration of substrate present in the sample. There's also some variability inherent to using bacteria in a test like this, so it's better to run this test at least in triplicate to make sure you have conclusive results. So here's an example. We were using an oxygen uptake test on QuickSign-P for bacterial degradation of whey protein isolate. Now, this is a test we were actually using to help establish our oxygen uptake methods. We had a whey protein isolate solution we made. We added a little bit of QuickSign-P to it, and we were monitoring how the oxygen uptake rates changed. This, unfortunately, we were doing manually. We had to collect data because we didn't have automated collection like we have now with the YSI probe. But anyway, the results looked pretty good, but what we were noticing is that we didn't have linear data for the first 5 to 10 minutes of the study. Non-linear data makes analysis more difficult in these cases. So a lot of the time, we actually look at 5 to 30 minutes in these tests and sort of ignore the first 5 minutes because it tends to have a lot of variability and a lack of predictability, which makes the test a lot less useful. And a lot of the oxygen uptake you'll see in the first 5 minutes has more to do with how the test was set up exactly than how the system is actually working. But if you cut out that time, you get a nice linear fit for the most of the rest of the graph. And you can see our R-squared values, which represent linearity, increase to nearly 1, which would be a perfectly linear fit, as opposed to the other ones, which we're looking at like 0.9, which is not awful, but it's not great. So this is a nice test we use a lot for a simple, does our enzyme work or not type test. This would be you'd add enzyme directly onto a solid substrate in something like a petri dish, or in this case, it was a 6-well plate. And you see, does the solid substrate break down over a period of time? Now, this is nice because analyzing results is incredibly easy. Either the substrate will break down or not. Usually, this test works well run overnight over a 2 to 3 day period. Setup is very simple. So this is a type of test you can easily do. The one caveat on this test would be I would make sure the pH conditions are favorable or representative of your system, so you know if a product is actually working in somewhat representative conditions. There are some limitations though. Solvents could have visual effects on fats, oils, and greases, meaning you could have a solvent product look a lot like an enzyme product. Frequently, you're going to end up applying a fairly high dose of enzyme product in this case because visual degradation relies on having a fairly large chunk of solids. If you have a large chunk, there's a much greater volume relative to surface area in the test, which means it's more difficult for the enzymes to access the entirety of the substrate, and you have to compensate this by adding a higher concentration in these cases. In addition, this testing is not necessarily helpful for dose rates, but I would say it's a good test to run on site if you have something specific you're interested in degrading, like a ball of grease sitting on top of your clarifier or something like that, or some paper you find maybe in your grit screen, something like that, which you can look at. You can take a chunk of it, add some of somebody's product, and see if it's actually breaking down in proper pH conditions. But you'll have to rely basically on literature dose rates in this case. So here's a really nice example of it. This was using QuickSign-P to help break down gelatin chunks. Gelatin is a very nice substrate for QuickSign-P. Not something you'll see a ton in wastewater, but it's a good one to test a protein because it tends to be very pure and very consistent. So we can see with the addition of 10 part per million of QuickSign-P here, we were seeing a visual degradation of the gelatin chunk, as opposed to our control, which essentially nothing happened in our period of this test, which is a 24-hour period. So there's some other good methods to analyze enzymes and bacterial products. A pilot scale study I really like. There's a lot of places which have some capability of running a small scale pilot system side by side. You can also commission other people to run this if you're concerned about it specifically. You can also scale this down to a lab study run in a longer term fairly easily to get representative results. But there you're looking for the specific impacts on the system. You can measure a number of variables and see if you're having positive impacts for a bacteria. But generally, I like to look for one specific goal for what you want to accomplish in the test. You can also run full-scale trials. That's simple, but you need a lot of baseline data in order to conclusively say if something's going to work or not. All right, so we have a number of products which we lump into the biocatalyst quality, which is a specialized blend of enzymes and bacteria in several cases. This is designed to function in wastewater, and they're targeted towards specific substrates which people frequently have problems with in wastewater. In particular, here we'll talk about Quiczyme P, Quiczyme L, bug juice, and Greasecyl liquid. We talked a little bit about Greasecyl already with our bacterial products. But Quiczyme L, the first one, this is designed to break down fats into fatty acids and long chain fatty acids into short chain fatty acids. So it's very helpful for grease degradation. It contrasts with Greasecyl, and that Greasecyl is designed for different time spans and conditions a little bit. So you wouldn't necessarily I wouldn't recommend using Quiczyme L in a lift station as opposed to Greasecyl. We've had some people try to use both in some cases. But generally speaking, Quiczyme L is designed to work in a longer time frame, and Greasecyl is designed to work in a shorter time frame. So Quiczyme L tends to work better in wastewater systems. Greasecyl tends to not do as good a job fully breaking down fats. So that would be a case where in your wastewater plant, we generally wouldn't recommend Greasecyl. There's also a number of ingredients which are not really designed in Greasecyl to be used within a wastewater plant, like an activated sludge system. They aren't necessarily harmful, but the efficiency might be reduced. So that would be how you'd kind of weigh these products against each other. But Quiczyme L will break down fats. Generally speaking, you can measure the effectiveness of this product with COD reduction, which is nice. We've produced some decent literature doing COD reduction on it. We recommend 10 to 60 degrees Celsius for a temperature range and a pH of 6 to 8 for optimal pH function. Now, with all of these products, generally, if you are above the temperature range, I wouldn't even mess with adding the enzyme. It won't persist for long enough to be of any value in your case. If you're below the temperature range, talk with us. It's possible if you have enough holding time that you can get it to work in your system. pH is similar. If you're a little below pH 6, or in the case of Quiczyme P in our next slide 7, you can still get some effectiveness of the enzyme. It will just typically be slower. Whereas above pH 8, I definitely wouldn't use Quiczyme L, and generally speaking, above any enzyme ranges of pH, I would tend to stay away from it because they will both function slowly and survive less time in a system if you're outside that range. All right, so quickly going through our Quiczyme L study at UW-Stevens Point. In this case, we were applying 10 gallons per 10,000 gallons as an equivalent dose rate. It was 20 degrees. We added a mixed liquor sample from a system, and we spiked fat soils and grease from a variety of sources into the sample, along with influent, to see how Quiczyme L influenced the breakdown of fatty acids. So we partnered with the University of Stevens Point for this study, and they provided us the data. This complete study is one of the attachments we provide. And a couple interesting things. Note, you'll see both with bacon grease and with milk fat at the end of the test, you're seeing a similar reduction in fatty acid concentration, regardless of which substrate is present. But if you look at the front end of the test, our bacon grease optimal separation between the two curves was at about 9 hours, and then all the points were essentially parallel between the two. There was not really there was not any decrease in performance over the period of time, but most of the effects were already observed at 9 hours. However, we didn't see great effects at about 4.5 hours in this case, whereas when we used milk fat, we ended up seeing much better results around 4.5 hour time point, and then everything kind of became parallel and we didn't see much change, indicating the enzyme did most of its work in the first 4 hours for this test in breaking down the substrate present. So be aware, we've tested this with a number of different substrates. If you're concerned about something specifically, I do recommend looking up this study, but generally speaking, the optimal time for Quiczyme L to react with the substrate will vary somewhat depending on what type of fat is present. So Quiczyme P is our next biocatalyst product. I mentioned a couple of these things already, but Quiczyme P is designed to break down proteins into amino acids. It can be really helpful in cases like dairy systems, which you have a lot of nitrogen in the form of protein that is a bit difficult for bacteria present to access. In those cases, you can apply Quiczyme P, and it'll assist in the degradation of protein so that heterotrophic bacteria are able to pick up those amino acids and use them as a nitrogen substrate. And in this case, it can limit the need to supplement something like urea in a plant where you have protein present, but it isn't bioavailable. Our temperature and pH conditions are a little different with Quiczyme P than Quiczyme L. Generally, it works a little better in slightly higher pHs, and the temperature range is similar. Just a note about it, don't mix it with Quiczyme L. It's best to add these far apart because Quiczyme P can actually break down other enzyme products if they're added together at the same point in time, or particularly mixed in a drum. So a lot of the time we recommend maybe adding Quiczyme P a couple hours away from Quiczyme L, or add it at a different point in the site. Maybe add Quiczyme L further upstream and Quiczyme P further downstream, that kind of thing. So both of the enzymes have an opportunity to work. All right, so we have a couple different data points on Quiczyme P. This study is representing total protein hydrolysis taking place. Degree of hydrolysis testing allows you to analytically measure how broken down proteins are with the addition of something like Quiczyme P. In this case, we saw, as you saw before, great results with gelatin. In fact, the breakdown time for gelatin was nearly tripled compared to the control, or over tripled compared to the control, I should say. Whey protein was almost twice as fast as the control, so we were seeing very effective results with whey protein. Egg whites, we did see a 29% improvement. That's pretty good. Egg protein tends to be a little bit inhibitory of protein degrading enzymes, so seeing some results we considered promising, but you can imagine in an egg waste plant, you'd want somewhat better conditions for Quiczyme P to work than something like whey protein and gelatin or other proteins. You may get much better results with these products in those cases. All right, so here's a bit more data. I already showed an uptake test with Quiczyme P earlier on with the whey protein isolate. This was a mozzarella plant, and this was kind of a really good case where either the system was having difficulty producing protease for some chemical reason, the bacteria were just struggling with this case, or there was a very high concentration of undegraded substrate present because we were seeing an 80% increase in oxygen uptake rate relative to the control. Generally, in an oxygen uptake rate, if you're above about a 10% improvement, you can start to be confident that you're actually seeing some improvement, but it might be more minor. It's a little hard to predict exactly how these will work in a system, but if you see like an 80%, that's a very promising case of your system is obviously having trouble degrading proteins, and clearly the addition of something like Quiczyme P is able to speed that up. All right, bug juice. This is more of an acidic pH additive. It typically is effective on both paper fibers and fat, cellulose, and grease to some extent. It has a similar temperature range to the other enzymes we have recommended in product or Biocatalyst products. So for some data on bug juice, this was one of the most tedious tests we have ever done, but it ended up with very interesting data to analyze. So we first applied 1% paper fibers to a mixed liquor sample and observed the effect on settling. This was actually a mixed liquor from a digester sample. So the sample with the paper fibers supplemented settled at about 50%, whereas without paper fibers, we were seeing a little below 40%. They were getting pretty good solid separation in this site at the time. So we can see the paper fibers contributed to bulking in this digester sludge, which indicates it would likely have a poor effect on dewatering in this system. So with that in mind, we produced a 1% solution of paper fiber slurry. We applied bug juice to it, and we observed it over a period of time at optimal pH conditions. This in case we applied it at 10 ppm as well. And we were able to see a meaningful and significant reduction in paper fiber length with the addition of bug juice versus our control. This test was kind of funny because we ended up running it by actively counting each of the paper fibers in the system and measuring each of them. And we were looking to see if there was a decrease in paper fiber length in the system, which involved looking at hundreds and hundreds of paper fibers to have a statistically meaningful sample size in this case. But you can see here, bug juice clearly had a shorter paper fiber length. This could imply that it will have better settling properties in the digester sample. We had a bit of difficulty running this test with a mixed liquor sample because the paper fibers can sometimes hide in the sludge, and they can be a little difficult to count and quantify that way. And we like to have a predictable amount of paper fibers because it allows us to have more of a consistent test setup. All right, so our final product I want to talk about for a few minutes here is Greasecyl liquid. We already discussed our case study earlier where we were seeing some TSS and BOD reduction from the collection system. But here, A lot of the time, we recommend this product for grease traps and lift stations for the control of grease buildup. It's generally effective in pH 6 to 8, but you have a little bit of a broader range there with Greasezilla than you'd see with something like Quiczyme L, which is more ideal for lift stations. It tends to function pretty well outside of that pH range, at least for 1 or 2 pH values. So this product tends to be more effective in warmer temperatures as well. That tends to be a trend with enzyme products. So we partnered again with Stevens Point on this study. We applied Greasezilla to some beakers, which we added a grease as a substrate. We tested a number of different greases to see how this product was going to work and compared them side by side using mass reduction as our metric for measurement. So this test was run over an hour period of time. We were comparing it to a generic solvent competitor just so we had a reference point. And we also compared it to a blank, which the blank is just distilled water. In this case, we're hoping not to see any results with distilled water here, which we didn't. So that was a good sign in the study. But here's an interesting thing. You can see how a biocatalyst product like Greasezilla might differ a little bit from a solvent product. In some cases, more than other cases. And in some cases, you'll see much better results with Greasezilla. And in some cases, they'll be closer together. But in our composite grease, which is a blend of coconut oil, lard, and butter, we were seeing greater performance with Greasezilla versus the competitor product in terms of total fat reduction. In coconut oil, it was a bit more of an interesting picture where the solvent actually worked a little faster for the first 10 minutes. And then after 20 minutes, the Greasezilla had caught up with it. You have a little bit of time for it to get to work since it's a biocatalyst product. And then after that point, our Greasezilla passed up the solvent product. So in some cases, you could see an application where maybe a solvent could work more quickly. But generally speaking, Greasezilla works similar in terms of speed. And it did work better than the solvent we were testing on in these cases for this study. So just a couple key takeaways. Bacteria are not enzymes, but bacteria work more slowly and enzymes work more quickly. So this is pretty big stuff. One thing you'll want to keep in mind is persistence in a wastewater system. This is true for both bacterial and enzyme products. The longer a product persists in the system, the less precise your timing needs to be when you're applying a product. So in the case of enzymes, if you have a very high temperature or some sort of unfavorable condition, you basically need to add them at the exact same time the substrate is present. Whereas if you have a bit of a cooler temperature and favorable pH conditions, you don't need to add them at the same time and you'll still get most of the effectiveness of an enzyme product. And this applies to bacteria too. If your bacteria are able to persist for longer periods of time, you're going to see better results with less precise dosing. Make sure whenever you deal with studies in these cases to have measurable goals whenever you apply a product in your system. If your goals are not measurable, then you'll never really know if you're successful or not. And you can end up falling into the trap of, I'm just feeding bugs into my wastewater system. I was interested to see at the poll at the beginning, nobody, we did get 2 at the end, they told me to comments. They told me to is a common one we encounter for enzymes and bacterial products as to why people are using them on the field. If you happen to have a case like this, you want to make sure that it's going to work in your system. Anyway, the other thing to note is enzymes require specific conditions to work well, but bacteria generally need to have conditions which are favorable enough for them to persist in the system. Are there any questions? All right, so I just saw a post from Julie. Can a product decrease microthrics or other filament levels? And yes, we generally work with Quiczyme L for the reduction of microthrics. It tends to work really well in conjunction with Foam Buster. The idea here is Quiczyme L will produce more fatty acids, and Foam Buster will add nutrients which complement heterotrophs in a wastewater plant in order to degrade the fatty acids are present. If you don't add a supplemental nutrient and you don't have enough nitrogen present in a system, you're not going to see as great a result in the reduction of microthrics with something like Quiczyme L because you won't have the heterotrophs present won't have as much ability to degrade the high carbon loading which comes with something like a fatty acid. All right, so the next one, real world results magnitude compared to lab scale. That is a great question. As far as in your system, you're going to have to look at a lot of environmental constraints to determine real-world results. We have a lot of full-scale data on these products, But ultimately, in your site, it can be a little unpredictable. I do like oxygen uptake studies to determine real world results because in an oxygen uptake study, you're getting an increase in rate and degradation. So you can kind of calculate it out and get a rough idea how it would behave in a system. Alternatively, if you want to be sure about real world results, something like an overnight or multi-day study where you have a similar retention time in a reactor to do a test with the addition of product versus without product, you can measure the variables you're specifically interested in and compare something closer to real world results. So you have a better idea of whether you add it in your system or not. So in those cases, you should expect similarity, but there's always going to be differences in the system which can affect, the reliability of the results somewhat. So ultimately, testing it in your system is the only way you'll be 100% sure exactly what your real world results will be. All right, are there any other questions? Well, thank you all for attending today's webinar. I hope you all have an excellent rest of your day. Please reach out to me if you have any questions that come up regard to any of these products or literature. I'll make sure to get back to you. Thank you.